Welcome to IVOL's new webinar series. We called that Bioscan Illuminating Biodiversity. So today we're launching this new webinar series to introduce you to researchers and collaborators around the globe who are working on the front lines of biodiversity science. Today, the title of the presentation will be Macrogenetics through DNA Barcodes, and it's presented by Dr. Leonardo da Porto. Leonardo leads the Numerical and Experimental Zoology Laboratory in the Department of Biology at the Università di Firenze in Italy. So he has led comprehensive DNA barcoding initiatives, studying the evolution of butterfly biodiversity through time and space, including creating a complete library for Western European butterflies. Leonardo established the first phylogenetic tree for all European species and an extensive data set of traits. He also maintains and is an author of the Recluster R package, which is a statistical tool for the spatial analysis of biodiversity patterns. So, without further ado, off to you, Leonardo.
Me to this webinar. And I go straight to the topic, talking about macrogenetics. And what is macrogenetics is a very recent, field of research which explore the pattern and predictor of intraspecific genetic variation, sort of macroecology at the intraspecific level. Typically, it involves the use of many taxa, broad taxonomic level, and mostly at the broad spatial scale. Of course, interspecific genetic diversity variation is a key biodiversity aspect because it reflects evolutionary history, biological connectivity, and of course, it's also involved in conservation because also adaptability and viability of population depends in part on this feature. And typically, it encompasses two different facets. Within population genetic diversity, which is the genetic diversity existing inside each population, and of course, the genetic diversity between population, which is the main topic, for example, of phylogeography. And of course, macrogenetics is mostly limited, and we are starting to make macrogenetics because we are obtaining big sequence data which are necessary for such wide, large-scale studies. In a few studies, there are de novo sequences dedicated to the study, but in most cases, we need to obtain data from a previous study, usually in repositories like BOLD GenBank, obtained by phylogeography, taxonomy, and DNA barcoding. And this are typically integrated with big trait data. So we interconnect the DNA aspect with the functional aspects of a living organism to understand which factors determine interspecific genetic diversity. But most of this data is available as mitochondrial DNA sequences. Nuclear DNA sequences are still less represented, and they are more hard to compile. And so mitochondrial DNA is the main marker used in macrogenetics so far. And since macrogenetics are still rather limited by availability of sequences, they are still dealing with testing very basic and relatively simple hypotheses in three main fields of research, mostly in phylogeography, in which we search for gradients of IGV, like the latitudinal trend, and the correlation with IGV and species richness, typically with mitochondrial, nuclear, and other kinds of markers, and functional ecology, where research understand if interspecific genetic variation is correlated with species traits to understand the mechanism that allow the emergence of this phenomenon. And of course, in conservation biology, searching for the relationship between the specific genetic variation and adaptive and conservation status of species, and also understanding how the human impact can affect this IGV. And there is a strong debate if mitochondrial DNA markers are good markers for conservation. And of course, there are many papers saying not. This is because, of course, in many cases, we consider that mitochondrial DNA variation is not strongly linked with the variance fitness of populations, and also that the capricious variation of mitochondrial genomes makes really weak the mitochondrial markers for conservation for the fact that they do not reflect genome variation. So usually, okay, we avoid using mitochondrial DNA for conservation biology. At least we reduce the importance, and we try to focus on phylogeography and on functional ecology. And what I will present today are analyses that I completed today in the morning, and so beware fresh paint, and is mostly quite unpublished data on very wide patterns at continental scale on phylogeography and functional ecology. First of all, some description very fast of the main measures using to measure mitochondrial DNA IGV. If I have a series of individuals belonging to different populations, of course, any dissimilarity matrix, distance matrix, this can be simply p distances, will be represented by some relationship, which are intra-population p distances, the red one, of course, and some that are inter-population p distances. And there are very famous and commonly used parameters to measure which is the general diversity. Haplotype diversity is a typical measure, but there are also measures to understand which is the spatial structure of the of the populations. Typically, this is measured by DST, which is the HT, so the total distances minus the intra-population distances, which is quite similar to HS, but since in some cases we count average, it is not perfectly agree. And GST, which is a dimensional, varies between 0 and 1, is standardized, and it is 1 when at least one mutation is not shared completely between two populations, and there is no genetic variation inside the population, to 0, when of course all the variation among population is in the same order of magnitude of the total diversification. Of course, we can see here that the HS, so the interpopulation p distances, are usually much more than the intrapopulation p distances, because, of course, in many cases, in some areas and population, only one specimen is analyzed, and so in these cases there is no data about the intrapopulation distances, which makes the computation quite complex. And the link is the link between within this measure and the traits of species are quite clear prediction in phylogeography. Here I have included the haplotypes, different colors, of course. In these pies are different haplotypes for Vanessa cardui, which is a very famous migrant of Europe. And of course, we can see that more or less the haplotype diversity in Vanessa cardui and Lasiommata megera is similar, but in Vanessa cardui, it is the genetic variation is expressed as intrapopulation variation, sorry, while in Lasiommata megera is expressed as interpopulation variation. So in the first case, in Vanessa cardui, there is a GST presumably close to 0, and in Lasiommata megera, the GST is presumably close to 1. And in literature, there are some papers, very recent, the last 2-3 years, where one of the most important gradient in diversity is tested also on IGV. There is this paper testing that tropical low latitudes area have a higher IGV in mammals and amphibians, and this paper testing the same in freshwater and marine fish, and of course both using mitochondrial DNA data, almost the only data available. The reason why there is a latitudinal trend is a very established theory in biogeography, because the higher climatic stability of tropical areas in some respect hamper the extinction of the species and genetic lineages, so it enhances richness. And of course, also the high temperatures are predicted to accelerate metabolic rate, generation time, and also mutation rate. So this is a very well-established pattern in biogeography, and now we know that it's also probably an established pattern also at the genetic interspecific level. But one of the rules of the macrogenetic studies is that the variance of IGV explained by predictors is in general very low. So there is a lot of unexplained variance in the observed genetic variation. And most of the variation is not explained by our predictors, say latitude or less, but by differences among species, some idiosyncrasy that, as we can see that now, in some species there is even no effect for latitude or positive, and in other groups instead there is a clear negative effect of latitude, and in other groups there are effects of climatic stability on determining a higher genetic variation, and in other groups there is no effect. And so moving to butterflies, to a study we published a couple of years ago, we can see how that if we go with the general, these are more or less 500 species of butterflies from Western Europe. If we look simply at the haplotype diversity, the traits of the species do not explain the variation, but the variation is better explained when is analyzed in its spatial structure. So the more the spatial structure, like in GST, is examined, and the more there are these very clear, rather not so strong relationship with the traits. Look, the direction is important. In fact, the species with using more generalist species have lower genetic variation, while species with bigger wings also have lower genetic variation, because they probably are less mobile, so they have small species, and specialist species probably as less mobile, and I would say that probably they have lower gene flow. Range size is a very well-known predictor of genetic variation, because of course the larger the range, the more possibility for species to diverge, and also cleaning allopatry. And also there is the possibility that a higher population has more genetic variation. But another explanation of this very low variance, explain the variance, is the incompleteness of sampling. These are sampling of the previous paper I showed you on plants, on mammals and fish, and you can see that in most cases only a few areas were sampled, like this green area, but which are almost the same, which are North America, Europe, and in some cases Southeast Asia. For the rest, there is no such very good sampling, which can actually reduce the power of the analysis. But also, there is this particular behavior of mitochondrial DNA that makes it really unpredictable. For example, there are common genetic sweep, which makes IGV depending on time since the last sweep, which probably erased most of the genetic variation over big areas. There are endosymbionts which strongly affect the diversity of mitochondrial DNA and the spatial pattern, of course. The population size, which is expected to be positively correlated with genetic diversity, has changed dramatically, probably since the onset of the interglacial, with the expansion and contraction of the species. So its fingerprint is probably lost. And of course, mutation rate largely vary among taxa. And this brought many studies telling that mitochondrial DNA probably is the worst marker to study genetic diversity. But we go and we insist with this marker, and we wanted to inspect the phylogeography of butterflies in two very well-studied areas for which data are strong, which is Europe and North America. And we tried to test three main paradigms of phylogeography. Of course, the latitudinal trend. Then we wanted to test if the genetic contrast among areas are established and maintained along the main topographic barriers like sea straits and mountains. And of course, the third has been that if species have been trapped in different, we say, glacial refugia, and then have diverged there and expanded during or contracted during the current interglacial until the barrier that they found, they should show coherent regions that are called zoogeographic regions, where more similar lineage should be present across species. So, well, to do this, we took advantage of two big papers and on Europe and North America. And for Europe, we also increased the sampling size to more than 30,000 of sequences with more than 500 species. And the distribution of the species, these circles are proportional to the species, is highly correlated with the richness of Europe, as you can see. So since the larger circle is around 150 species, and the maximum is over 190 species for a place, we think that we have sampled almost all the diversity of species across Europe. And the gradient in genetic diversity was already found in our previous paper, where we showed that the number of haplotypes is much higher in southern part of Europe and then decrease, mostly in the areas that were covered by ice during the Pleistocene. But now we have to understand the two different possibilities for a latitudinal band to be richer than another one, which is the possibility to have in any point more haplotypes, because there is more intra-population diversity, or because in this band there are more populations structured like we have to remember that Earth is spherical, and that areas at low latitudes have a higher distance between individuals, which makes possible that this relationship between latitude and the genetic diversity is due to isolation by distance. So we proceeded like dividing Europe and America in bands and selecting the same number, minimum number of specimens per each band, so just to avoid that more specimen in one band, of course, increase diversity. We measured the distance between the barycenter of the specimens and all the specimens to control for the distance. And then we divided the mean genetic diversity, the mean p distances among the specimen of the same band. We standardized and we multiplied this for the GST of the species over the entire area and for 1 minus GST. What means if I multiply by GST, which is between 0 and 1 is for species with a full spatial interpopulation diversity. This means to weight the species, and in this measure I will have mostly the diversity which is interpopulation. If I multiply by 1 minus GST, I will obtain a measure which is mostly due to the species making interpopulation diversification. This is because I cannot work with intra which HS, because I have no enough data for this, despite the very big sampling. So I divided all the analysis between one analysis mostly based on species making differentiation among population, and one analysis mostly based on species that are mixing their population. And this is the result for, for latitude. Of course, the interspecific genetic variation has the latitudinal trend, which is very strong, together with the distances which are so controlled. But we can see that also using the functional trait of the species, phenology also determine a generally higher IGV. But what is more interesting here is the interaction between the band, so the latitude, and phenology and wingspan for IGV inter, and between wingspan and endosplant with latitude for intra. It's also interesting that both the two kinds of species both show the same trend. So both the species with making mostly diversification within population and those making difference between population, they both show the latitudinal gradients. So there are more haplotypes, more genetic divergence in a single population, more in the south, and also more population which are differentiated in the south. The pattern of the interaction is interesting and for me unexpected. I don't know really how to explain it, because it seems that the larger species have, this is due to the fact that larger species show a higher influence of latitude respect to smaller species, which is the red one. And this seems mostly because it seems that in the southern region, the bigger species have a higher genetic diversity in both cases, both in inter and intra population. The same for the North America, same pattern, same relationship both with distances and latitude. Here I could also try to reduce the data set from a very small percentage of 10%, 20%, 30% of samples random, and then to see the variance explained, which is still low because it's some 0.8%. No, yes, 8%. It's not very strong, but if I ratify the species, I can see that more or less is the same. So this means that probably is not sampling size, because if I would expand my sampling size, probably this trend remain the same and the percentage of explaining the variant is the same. So probably it's not due to incompleteness of the sampling, but is a phenomenon. Latitude explains a very partial variation of IGV, more or less 1%. And now we move to the genetic contrast. And in a series of 2 papers, we show that over Europe, over the Mediterranean, West Mediterranean, smaller and smaller, and the phonistic contrasts are concentrated along the seaside, but also that most of this contrast are due to cryptic taxa. So taxa that are in general phylogenetically similar, very close related, and so there is room for macrogenetics to enter this field of research. Of course, to do this, we carried out series for each species a genetic landscape, making the Leonid triangulation, calculating the path, connecting the locations, measuring also the maximum altitude and the minimum altitude encountered over the path, then making residuals with distance to avoid isolation by distance, and then making a map for each species, which has been averaged all over the continent. So we will obtain a single map. I go to the analysis where we can see that if we compare the genetic distance at the front of between specimens, the distance, of course, there is higher distance separate higher genetic diversity. Phenology also shows again an importance, because species that fly longer tend to have a lower genetic diversity at the end of each path. But what is interesting is that the path that cross higher mountains have higher genetic diversity at their ends. And also, when the minimum altitude instead is under analysis, we can see that path passing through the sea necessarily, and the deeper the sea, the stronger the genetic diversity in between. This is expected, of course, but is something that has been found all over the European butterflies and is sort of general result. And this is the pattern. This is the contrast, the map of the contrast for all the genetic diversity, and we can see that there are areas all around the sea which have strong genetic diversity at their sides. There is also some genetic diversity here in the very north, but if I make a partition between species showing genetic diversity intrapopulation, those showing interpopulation, the pattern changes, and I can see that the contrast in the north are limited to the species making genetic variation within population. If we go in the species making genetic variation between population, I can see that this contrast in Northern Europe almost disappear, with a very nice concordance with the distribution of the ice sheet during the Pleistocene. This is of course expected that the area have been recently invaded by the species by gene surfing, thus reducing the difference. But this is a bit more unexpected. And of course, these the location of this contrast agree also quite nicely with the expectation from Hewitt, who has drawn the main suture zone from Europe. Why this happens? Why mostly? I was really interested by this phenomenon, and so I went to the single species to check, and I found that there are many species that in the north show a sympatric population, almost sympatric, with strong interspecific, strong genetic variation. So there are lineage haplotypes, some 5.8% distinct that are completely sympatric, or 3.5 like in these other species. There are several also in the south, but they are mostly separated, and this pattern is never reversed. There are never species that are highly differentiated in the north, which show a single lineage in the south. So we must remember these two facts: the ice sheet and the fact that this is a fake map, because this area is not large, are this one. And what happens what can be the reason for this? It's possible that the species that are adapted to very warm climate are those that survived the Pleistocene in North Africa, which is a single area. And this probably have invaded Europe quite recently in a single haplotype. So I could expect that these species have a very low haplotype diversity, very low GST. But the species that have quite lower thermal preference probably have survived also in the distinct refugia, which makes them to expand just a bit during the interglacial, and creating this very strong contrast area in the Mediterranean. But if I go with species more and more tolerant to cold, they should move far to the north, and at the end they should mix. And when they mix, probably, for sure, they will reduce their GST, but maintain their haplotype diversity, because there are still 3 lineages, but they are mixed. The more I proceed, probably the population to the south do not occur in species. These are different species, of course, in species that are more tolerant to the cold, until the very cold species, of course, will have to live in the areas that were previously occupied by ice sheets, but they will coexist because they are only very small areas where they all converge, and they have to decide or to coexist in sympatry or not to go there. And of course, this probably has created this pattern of intrapopulation IGV as compared to the interpopulation IGV in the south. The same occurs in North America. This is the overall pattern. This is the interspecific pattern, where the areas that were covered by ice still have some genetic diversity, which is almost lost for interpopulation genetic variation. Of course, again, there is a strong relationship with distance, and a good relationship with maximum altitude encountered along the path connecting two populations. And also, these are the classic suture zone identified by Remington, and there is some correlation with these suture zones. Some are quite clearly retrieved, some are just a bit moved to the south, but I would say that there is some nice correlation. And now we move to the last analysis, which is the geographic region. The geographic regions have been, we tried the first regionalization based on genetic diversity for Southern Europe, for where there were good sampling, and we obtained these maps. But of course, we wanted to improve the method. This is a bit complex, but basically it produced among the units sort of a dissimilarity matrix based on the average of all the species available. And of course, based on this dissimilarity matrix among the spatial units, we simply make a dendrogram, and we cut the dendrogram at different levels, thus obtaining a different cluster. And this cluster explain part of the dissimilarities, which means that which is the dissimilarities within a cluster compared to dissimilarity within clusters. And the first division in two areas, of course, explain very small part of the genetic variation among units. But if I go with a higher number of clusters, of course, the variation explained is higher, and until I arrive to a value of around 90%, which in zoogeography is considered a very good level of explained dissimilarity. Not yet, but now this is more than 90%, and here is we can see how the main so-called glacial refugia are highlighted. There is the Iberian refugium, there is the Italian refugium, there is the, say, Balkan refugium, there is Turkey, there is of course also North Africa, which was not considered by Hewitt. And I would say that here the correspondence between the suture zones identified by Hewitt and the boundaries among the areas is quite strong. We can go deeper in creating more and more areas, but now each division increase only a bit. They explain the variation, so around 1%. So maybe these sub areas have not such very great importance. What is interesting is that was the regionalization you made based on interpopulation IGV. This is instead what I obtained with intrapopulation. We can see that still there are the main peninsulas in the south, but in the north, again, the pattern is completely mixed. Probably because there is this confusion of sympatric lineages that make, well, simply make confusion and cannot provide a clear division in areas. Again, for North America, well, let's arrive to the 90% straight, and this is, we obtain these seven areas with two eastern and a group of the central regions and two regions in the northwest. And again, there is a nice correspondence with the Remington suture zones. Just if we just move a bit to the north, this or this a bit again must be moved a bit to the north. There is also this distinction and this. So I think that most of the expected pattern based on just on comparing different species has been confirmed based on numerical analysis on all the butterflies existing in North America. And if we go with intra-population variation, of course, the pattern is no longer spatially coherent here. But this is expected because, of course, we don't use the species that make differences among areas. So, in conclusion, we show that macrogenetics based on mitochondrial DNA has confirmed the three main paradigms of phylogeography over Europe and North America, using an interior taxon of species. So maybe this marker is not the bad, as in some cases is expressed. And well, however, we can find that the variance explained is low, and probably this is not due to the to sampling incompleteness, but is really due to differences among species in something that can even catch by functional traits, because also adding functional traits, the variance explained is still quite low. So which are the future challenges of macrogenetics through barcodes? This is what I try to write down my hypothesis. One is define the taxa. I started all my analysis without telling how these taxa, where we measure intraspecific genetic diversity, are identified. This is just taxonomic criterion. It's just that somebody, specialist, telling what is a species and what is not, which is like to set up a cut over the continuum of speciation, which divides macroecology from macrogenetics. This is, of course, the elephant in the room of macrogenetics. Imagine this species. This species is an endemics from our Italian islands, which is considered as a single species in the recent literature, but has a divergence higher than 5% in mitochondrial DNA among population. So if I use only one species, as I'm doing now, I have one species with a very strong IGV. But if I divide the species in three taxa, I will have three taxa living in the south with very low IGV. And this changes a lot the conclusion and the analysis, because this is one. But if we change for many species from splitting to lumping taxonomy, this probably will change the results of macrogenetics. Also, we know that different specialists in different taxa are used to separate species with quite different levels of divergence, and this could be also the reason why there are different results for different taxa, the idiosyncrasy among taxa. Because if we look at, if we divide species at different stages, we are looking at different times, and the process that have operated at different times, and probably also different processes. So in this case, mitochondrial DNA can help also because it can we can produce with mitochondrial DNA some division which can be more objective, like with some GNYC or other species delimitation approach. But the other point is possible to project macroecology, macrogenetics with mitochondrial DNA into conservation? Well, there are recent papers, reviews about macroecology and conservation, and they say that the main contribution of macroecology to conservation is identified by diversity hotspot, recognizing the effect of human activities also on climate change, on changing the diversity, and on developing methods, mostly to move from correlative to causal analysis. But if this can be done in macroecology, can also be done at the level of macrogenetics on evolutionary significant units. I can't see any reason why this boundary should prevent us to work with conservation. Another point very important for to project mitochondrial DNA macrogenetics in conservation is to understand if and to which level the mitochondrial genetic variants show difference in metabolic performance. There are some papers in some species because if the genetic sweeps are so common to determine a large part of the fraction of the mitochondrial DNA variation, then this means that they have a very strong adaptive value. And so not only the movement of the species to the north, but also moving of the genetic lineages to the north can be fundamental to maintain the observed diversity. Imagine that in southern areas, we have a higher number of haplotypes, and these haplotypes are probably adapted for the respiration in warming areas, and these which are probably the climates that will be in the future in other parts of Europe. So preserving the genetic diversity also of the mitochondrial DNA, and to facilitate this spread, the normal spread, the normal sweeps, maybe will become an important challenge for conservation, but really we don't know yet because there are not so many studies highlighting the importance of variants of the mitochondrial DNA on metabolic performance. And well, the last one is, of course, recognizing the effect of endosymbionts like Wolbachia, another elephant in the room, because it's possible, it's really possible that most, large part of the unexplained variants in HIV is due to this infection of these endosymbionts. And understanding this phenomenon and the possibility to block dispersal, because if endosymbionts, a population infected blocks the spread of another one, can be important with respect to the possibility that this sweep can occur or not. It is a very strong effort sequencing also Wolbachia, other than mitochondrial DNA, but probably is worth it. So I close here. I hope to have provided some nice view of phylogeography with macrogenetics, and I'm thanking my main collaborators for the American side, Paul Ebert, Jacopo d'Ercole, and from the European side, the group of Roger Villa, Gerard, that luckily are more serious than as they appear in this picture. And of course, Vlad Dinka, Luca Voda, the group from Emilio, and my colleague from Italy. Thank you.